The first thing you need to know is the structure. So basically, nucleic acids are composed of three things. So first, there's the ribose sugar, there's a nitrogenous base, and there is a phosphate group. And so the way these come together in the monomer form is called a nucleotide. And so the way that works is that you have our ribose sugar. So it looks a little something like that. It's this little hexagon-shaped thing, or not hexagon, pentagon. And then you have a nitrogenous base up here. And you have a phosphate group up here. So this is the very basic structure. We're just going to call this N for now. And this is the very basic structure. And the way this works is you have these carbons that are labeled individually. So this would be our 1' carbon, this is our 2' carbon, 3' carbon, 4' carbon, and our 5' carbon up there. So this isn't super useful for the actual structure, but it's going to be really important when we start talking about nucleic acids, so DNA and RNA. And those are super important because basically everything that makes you, it comes from just all of these, a ton of these little guys. So that's why that's super important. So let's take a look at what happens when we put these together. So I'm just going to get rid of these for simplicity, but just those are still there. But when we take multiple of these, we link them together. And what happens is it ends up looking a little something like this. There's a bond between this phosphate group down here and this 3' carbon down there. And basically what we can do is we can actually link a ton of these together and make these super long chains. And so this is a nucleic acid. So let me actually move this so it's a bit easier to see. So this is what makes a nucleic acid. So this is one strand. And so these things right here, this right here is the backbone of the DNA. And so the way this all comes together is you take two of these strands for DNA. This is for DNA, and we'll talk about DNA versus RNA in a second. But basically, there are these two things, and the way these work is these are anti-parallel, which means it's two strands. We have one going up like this, and one going down like this. So they're parallel, but they're just facing opposite directions. And so the way we can categorize this is up here, we have this 5' carbon. So this actually gets called the 5' end. And down here, we have this 3' carbon. So this gets called the 3' end. And so it's the exact opposite on the other side. So, for example, we have this 5' carbon down here and this 3' carbon up here. So if anytime somebody refers to, oh, it's 5' to 3', that means starting over here and going this way. So that's not super important for this unit because this unit is all about the structure. But these, so this is a nucleotide. That's one individual. But now this is a nucleic acid. So now the question is, we have all of these guys. So what is that? So what those are is those are nitrogenous bases. So we're going to talk about just DNA first. There's a little bit of variation when it comes to RNA, but this is basically the main thing. What you have A, C, G, and T. And these are your nitrogenous bases. These bases all stand for something. For example, the T is thymine, but you don't actually need to know the names, but what you do need to know is how they go together and how you can classify them. So first off, how they go together. So base pairs is how you arrange these things. So certain bases go together. So, for example, if this is an A, this is a T. So we can put these rules over here. Basically, A and T's go together, A and T's go together, and C and G's go together. And so that's how you would pair it. So, for example, if we just take this, for example, if we said this is a C, this is an A, this is a T, and this is a G, well, then we know what these are because we know what pairs with the other ones. So if this is a C, then we know this is a G. If this is an A, we know that's a T. If this is a T, we know that's an A. And if this is a G, we know that's a C. Great. And so now, how do we classify these new nitrogenous bases? So we have two things. We have purines, and we have no, that's not how you spell it. Pyrimidines. Great. So pyrimidines, a way you can remember this is PUGA2. PUGA2. So this stands for purines. This is just G, this is A, and the 2 represents the structure. So the way these nitrogenous bases work, it's either a 2-ring structure or a 1-ring structure. So for purines, they're actually a 2-ring structure. So this would look something like this. We have a hexagon, and we have a pentagon attached to it. I know that's not like a great drawing, but that's how it is. It's 2 rings together. And so this is A and G. And then pyrimidines, there's not really a fun name for that, so you're just going to have to remember that. But those are just 1-ring structures. So this is a 2-ring structure, 2 rings. So this is a 1-ring structure. So we can just draw our hexagon like this. This is C and T. And this is a 1-ring structure. So the way this works is you'll notice is one purine pairs with one pyrimidine. So if we look over here, we have C pairing with G. Well, C is a pyrimidine, and G is a purine. So that's how they kind of pair together. So you always have one pairing with another. And one final thing is hydrogen bonding. This is pretty niche, but it's still helpful to know. But these two sets of nitrogenous bases actually make a different amount of hydrogen bonds. So the way you have DNA, you have a structure. It's a double helix, and it kind of looks like this. And then you have these ladders. If you've seen any sci-fi movie ever, they love showing you this. Jurassic Park, all that good stuff. You see this ladder. So where does this ladder come from? It actually comes from hydrogen bonding between the nitrogenous bases. Those are pretty bad drawings, but the point still stands. Those are just hydrogen bonds there. So how many do these make? Well, A and T make 2 hydrogen bonds, and C and G make 3 hydrogen bonds. If I was to remember, C and G rhyme with 3, and I like to think like A and T to both take like 2 strokes to draw. Like whether it's A, it's like 1, 2, and T 1, 2. That helps me remember, but really whatever makes it work. So now we need to talk about DNA versus RNA. So we have DNA, RNA. So for DNA, this stands for deoxyribonucleic acid. And so basically, these have the nitrogenous bases A, C, G, and T. That's what we talked about. And basically, what you'll notice is if we actually take a look at one of these groups, what you'll notice is here you have an OH, but here you only have an H, as opposed to RNA. RNA will have this OH. And what's important is that this makes it much more stable. So that's why DNA is much more stable. That's why DNA doesn't fall apart where RNA really falls apart, and that's why it's like more temporary, but DNA is always there. So that's why DNA, that's super important. That's why you really want to make sure your DNA is okay. And RNA, that's just ribonucleic acid. And so that had slight difference. Instead of A, C, G, and T, it's A, C, G, and U. That's the difference, that U versus that T. For the function of pairing, it's basically the same thing. A U is basically just a T. That really comes into play in Unit 6, but U is basically just like the T equivalent. It pairs with A, and it makes 2 hydrogen bonds when it's paired. And so this actually falls apart. So if we actually take this structure and move it over, this has that extra oxygen. And so that's why it's really important, because that makes it a lot less stable. And so that means it falls apart more easily, and that's why you're constantly making new copies of RNA anytime you need to make a protein. Alright, here in post-production, because I totally forgot to mention this in the actual video, but basically RNA is a single-stranded molecule. Where you see DNA, you have the double-stranded double helix with the ladder. RNA doesn't have that. It's actually just the one strand that goes down the middle. So that's a really big key difference, and you should definitely keep that in mind. Alright, so that's it for nucleic acids. Here's a picture of the screen if you want to screenshot that for later reference. And I hope we'll see you in the next video.